And it is now my real pleasure to introduce Geneviève Deblois, our next speaker. Geneviève is a new investigator at the University of Montreal. She started in March 2020. She's an assistant professor in the Faculty of Pharmacy, and she is a principal investigator at the Institute for Research in Immunology and Cancer, or IREC. The main focus of our lab is to study microenvironmental heterogeneity and its involvement in the development of drug resistance, particularly in breast cancer. Her team uses metabolomic and genomic technologies to understand the reciprocal regulation between cellular metabolism and epigenetic processes in cancer cells. So Geneviève, without any further ado, the stage is yours.
Thank you, Marco. I'm just gonna select my pointer. Okay. So I hope you can see the slide good. So thank you very much for this introduction. I also want to thank the organizers for this invitation. I'm very happy to show our work here today. So I'm going to be talking about metabolic and epigenetic adaptations in breast cancer and how these adaptations can lead to vulnerabilities that can be exploited for to stop growth of breast cancer. So as Marina has already introduced, breast cancer is a very heterogeneous disease, and our lab focuses mostly on the triple negative subtype that overlaps quite significantly with the basal-like subtype. This subtype, as Marina said, is ER negative, PR negative, and HER2 negative, so it doesn't express any targetable markers, and these patients are currently treated using chemotherapy. However, resistance in these patients is very common, and there's a need to improve treatment and better understand mechanisms that lead to resistance. So as I said, we are interested in studying the progression of breast cancer, specifically triple negative breast cancer, and how they can become resistant to therapy by changing their cell identity. And our lab specifically is interested in how this cell identity is modulated by changes in cell metabolism and epigenetic reprogramming, which are two events that can regulate each other. And that's what I'm going to discuss today, and how their mutual regulation or reciprocal regulation can change cell identity to allow adaptation of tumors and allow progression. So I wanted to start just by showing you quickly some of the results that I produced during my postdoc when I was a postdoc in the lab of Mathieu Lupien, just to show you how we came to this idea of studying the reciprocal regulation between metabolism and epigenetic in tumors. So when I was in Mathieu's lab, what I wanted to do was to understand metabolic adaptations that were going on in drug resistance and how they could affect the epigenetic landscape of the tumors and allow tumors to become resistant to drug treatment. So we developed triple negative breast cancer cell lines and PDX models in collaboration again with Morag Park's lab at McGill University. And we looked at their metabolomic reprogramming. And what you're looking at here on the left is a volcano plot that's representing the metabolites that were significantly altered in the resistant taxane or paclitaxel resistant tumors compared to the sensitive tumors. And on the right, you have the pathway enrichment or metabolic pathway enrichment that these metabolites or altered metabolites belong to. And I got very interested in the cysteine and methionine metabolism that was significantly decreased in the resistant cells. And the reason for my interest in that pathway is that the methionine cycle produces a metabolite that's called SAM, S-adenosine methionine, which is the methyl donor for methylation reactions in the cells. So all the methylation reactions need SAM. And what we noticed is that our resistant cells show a significant decrease in the level of all the metabolites of this cycle, including SAM. So then we investigated whether this was affecting the methylation profile of the cells, and we started by looking at DNA methylation. So here is just a representation showing you how metabolites can affect epigenetic enzymes. There's a lot more metabolites that can contribute to post-translational modifications of histones. But you can see here some that's being used by DNA methyltransferase and histone methyltransferase to add methyl group on either on DNA or histone tails. So when we looked at DNA methylation in those resistant cells, we observed a significant increase in the number of CPGs, or the number of regions that lost DNA methylation. And we found that these regions that lost DNA methylation in the resistant cells were located mostly in intergenic regions. We also observed, there's a typo here, sorry. We also observed this in taxane resistant PDXs, which showed that as the tumors were treated, they lost DNA methylation as they became resistant. We next profile different histone marks to try to see whether we could observe a trend of histone methylation change in the resistant cells as well. And the only mark that we found that was significantly changing the resistant cells was the H3K27me3 mark. And as you can see here on this genomic representation, you can see that the resistant cells show an increase in H3K27me3 marks at very wide regions over the genome. Some of the peaks, the single peaks, were actually lost in the resistant cells, and the overall intensity was not significantly changed. So it was really a reorganization of the profile. But when we overlapped it with the DNA methylation loss, we observed that these increase in H3K27me3 region were significantly overlapping with the loss of DNA methylation, as you can see on this figure here. The fact that these were happening in intergenic region led us to investigate whether they would regulate non-coding RNA, and we indeed observed that these regions were enriched for specific families of transposable elements that are able to form double-strand RNA when they are expressed. So we investigated whether we could inhibit this H3K27me3 mark to re-express what's in these regions, or the non-coding RNAs that are in these regions. And we used an antibody that specifically recognized double-strand RNA, and we showed that upon inhibition of EZH2, which is the methyltransferase that's responsible for the H3K27me3 modification, we observed a significant increase in double-strand RNA, but only in the resistant cells. And this is presumably because the parental cells had this protection with DNA methylation. We also interrogated RNA-seq data in parental and resistant cells to identify whether they show an enrichment of the signature for viral mimicry or interferon response, since double-strand RNA is able to induce this signature, this gene signature of interferon response. And what we observe is that the initial paclitaxel treatment was actually able to induce this viral mimicry signature, but that this was significantly downregulated in the resistant cells, and that the treatment with UNC1999, the inhibitor for EZH2, was able to reactivate this signature. We also tested whether we were able to impair growth of the cells, the resistant cells and the tumors, using the inhibition of EZH2. So here we're using the UNC1999 as well as the GSK343. And you can see that while treatment did not affect the parental cells, it did have a significant effect on the growth of the resistant cells. We observed the same thing on the TNBC PDXs that we had that were resistant to paclitaxel. So you can see that in parental cells, we had no significant effect of UNC alone. However, in the resistant cells, we're able to slow down the tumor growth. And you can see here the survival time that's increased. So this led us to a conclusion of this project, where we observed that the initial drug treatment with paclitaxel is able to induce double-strand RNA. I didn't show this data, but it's the treatment itself is inducing it. And it might or not contribute to cell death. But what we observed is that upon development of drug resistance, this induction was inhibited, and the cells adopted a different metabolic profile in which they changed their methionine metabolism. And this led to a decrease in SAM availability that was reflected as a decrease in DNA methylation and a reprogramming of H3K27me3 to compensate as an epigenetic switch mechanism to compensate over those intergenic regions. And next, we showed that we're able to use an inhibitor of EZH2 to disrupt these regions and re-induce expression of double-strand RNA to induce viral mimicry or interferon response, which leads to an epigenetic vulnerability in these drug-resistant cells. So based on this, when I next moved on to my own lab, I wanted to pursue this study of the epigenetic reprogramming and cell metabolism reprogramming. That's the relation between those two adaptations and how they could impact cell identity. And the next data that I'm going to show you is actually looking the other way around. So we're looking at how epigenetic reprogramming is affecting cell metabolism in cancer cells and how we can use these adaptations as vulnerabilities to stop tumor growth. So I was interested by the observation that we did during my postdoc, that the treatment with the EZH2 inhibitor had no significant effect on the growth of the sensitive PDX or the sensitive TNBC cells. And I was wondering whether the resistant cell had adapted their metabolism so that they become more sensitive to the treatment with UNC. So I decided to look a bit more into what the actual EZH2 inhibition treatment was doing to the cells, or whether it was doing anything. And when we profiled the RNA-seq or gene expression profiling in TNBC PDXs or TNBC cell lines treated with an EZH2 inhibitor, the UNC1999 in this case, we observe a significant number of metabolic genes that were actually induced and pathway profiling led us to observe an enrichment of specific metabolic pathways, including cholesterol homeostasis and amino acid metabolism and fatty acid metabolism. And I got interested in the amino acid metabolism in part because of the project that Marina just presented before that was already going on, and where we saw that inhibition of EZH2 was able to induce GS and induce glutamine metabolism. So we started looking at other genes in amino acid metabolism, and we noticed that some of them were actually regulated at the transcriptional level directly by EZH2 inhibition, and showed a peak or a enrichment of H3K27me3 at their promoter, but a lot of them do not show a peak. So we're thinking that there is another mechanism that's probably serving this transcriptional effect upon EZH2 inhibition. So the first thing we wanted to look at was how the cells reacted or changed their metabolic needs or metabolic dependency upon inhibition of EZH2. And to look at this, we measured the amount of glutamine and glucose and other amino acids or other nutrients from the media that are taken up upon inhibition of EZH2, either taken up or released. So you can see when we look at glutamine uptake, and glutamine metabolic pathway is illustrated here in purple, we can see that inhibition of EZH2 led to an increase in the uptake of glutamine. So when you see the bar here below, it's an increase. So it's increase of glutamine uptake upon inhibition of EZH2. When we looked at glutamate release, however, it's increased, and glutamate is usually released as a mechanism to uptake cysteine, and cysteine is used in cells to produce glutathione upon oxidative stress. So we can see the increase in glutamine uptake and the increase in glutamate release. However, when we looked at glucose uptake, there was no very significant effect, and same thing for lactate, except maybe in one of the cell line where we saw a small increase in lactate secretion. We also looked at other metabolites from the media, and we noticed that a lot of metabolites were also, a lot of amino acids in particular were also taken upon inhibition of EZH2, especially amino acids that belong to the branch chain amino acid like leucine and isoleucine. And that was interesting because we observe an enrichment of this pathway upon inhibition of EZH2 in the triple negative breast cancer cells. And it's, we also observed that some of these genes that are upregulated show a peak of H3K27 and their promoter, but again, not all of them do. And there was also a study that was published in 2019 that shows the loss of EZH2 that reprograms the branch chain amino acid metabolism to drive leukemic transformation. So we next looked at intracellular metabolites and how they behaved upon inhibition of EZH2. So looking here at pyruvate, which is the end product of glucose, we noticed that there's a significant decrease in the level of pyruvate upon inhibition of EZH2. We next noticed that the level of alpha-ketoglutarate, which is a product of glutamine oxidation, is also decreased upon inhibition of EZH2, but glutathione is increased. So you can see that glutathione here, which is probably being used to detoxify the cells, is increased upon inhibition of EZH2. We next look at metabolites from the TCA cycle, which is usually used for producing energy, and we didn't see a significant change in the steady state level of these metabolites. So because of that, we wanted to understand whether the level of metabolites that we were observing were increased or decreased due to being used more or being less produced. And to do that, what we did is metabolomic tracing. So it's a stable isotope tracing of glutamine that's labeled on 5 carbons. So all 5 carbons of glutamine are actually carbon 13, which increases the mass of glutamine and allows us to detect incorporations of the carbons within the different pathways. Using mass spectrometry. So by doing this, we first observed that the amount of alpha-ketoglutarate incorporation of carbon 13 in alpha-ketoglutarate was significantly increased. So although alpha-ketoglutarate was decreased at the steady state level, its incorporation or its biosynthesis is increased upon inhibition of EZH2, which means that it's probably being made a lot, but also being used a lot. We observed the same thing for glutamate, and we didn't see anything significant for succinate. which indicated to us that glutaminolysis is increased upon inhibition of EZH2. When we looked at the metabolites of the TCA cycle, that became very interesting. So what we observed is that when we looked at the oxidative TCA cycle, so that's the cycle when alpha-ketoglutarate comes in and goes in a clockwise direction, we observed a significant decrease of most of the incorporation of carbon-13 in most of the metabolites, or actually all of the metabolites that we detected. However, when alpha-ketoglutarate is going in the TCA cycle, it can also go in reductive carboxylation. So that means it goes the other way around, and we can detect the difference by the different mass that is produced from glutamine. And by doing so, we noticed that these metabolites were actually increased, or the incorporation of carbon-13 was increased in reductive carboxylation, which indicated to us that the inhibition of EZH2 is actually decreasing the oxidative function of the TCA cycle, but increasing its reductive carboxylation. To test that further, we looked at oxygen consumption rate, the OCR, and the extracellular acidification rate, the ECAR, which is representative of oxidative phosphorylation or glycolysis, respectively. And we did not observe a significant change in the cell lines that we looked at upon inhibition of EZH2. However, when we challenge the cells by blocking different components of the mitochondrial respiratory change, we observe a significant increase in maximal respiration capacity of the cells, which indicates that they can respond better under stress or respond to increased energy demand upon inhibition of EZH2. And we think that this might be happening because some genes seem to be directly regulated by EZH2 or H3K27me3 LOCKs. Those are genes that are part of the TCA cycle. I just want to mention something quickly about reductive carboxylation. So it's regulated by mitochondrial NADH redox state, and it uses NADH shuttling couples cytosolic reductive carboxylation of glutamine with glycolysis in cells with mitochondrial dysfunction. MDH1, which is the enzyme that forms malate, couples the reductive carboxylation to glycolysis to support cell migration. So it's a pathway that's being used to support cell migration when there's a metabolic defect in some of the other pathways. It supports cancer cell in hypoxic condition, and it supports de novo lipogenesis reaction. And this was very interesting because we actually observe a lot of these metabolic pathways related to lipogenic metabolism that are upregulated upon inhibition of EZH2. And some of them, again, show marks of H3K27 of their promoters, but not all of them. So finally, what we did was to look at how these cells react to changes in availability of some of these metabolites. And of course, we looked at glucose and glutamine, which are the two main metabolites that feed cancer cells. And we were surprised to observe that upon inhibition of EZH2, we actually observed that the cells are doing better in low glutamine than the untreated cells. So cells are usually very sensitive to glutamine deprivation, but the inhibition of EZH2 seems to allow them to survive better this deprivation of glucose. We next look at glutamine deprivation, and we observe the opposite, which is in line with what we observed earlier, the highest dependency on glutamine metabolism upon inhibition of EZH2. Likewise, we used drugs to confirm this. So by using a drug that inhibits glucose uptake, it inhibits GLUT1, which is the transporter for glucose uptake, we observed that the cells are actually less sensitive to this drug upon inhibition of EZH2. However, when we use drugs that inhibit pathways in the glutaminolysis or glutamine metabolism, or generation of glutathione, we can see that the cells become more sensitive to these drugs when they're treated with an EZH2 inhibitor, either the UNC1999 or tazemetostat. So in conclusion, what I showed, especially in the last part, is that the TNBC cells with low EZH2 activity can express high GS, and this is in link with Marina's project, and glutamine metabolism enzyme. They show an increased glutamine and other amino acid uptake. They are more sensitive to glutamine deprivation. The inhibition of EZH2 activity can increase reductive carboxylation in TNBC cells. This is used for metabolic adaptation. We're also investigating whether it increases lipogenesis and cholesterol metabolism, fatty acid oxidation, as well as butyrylation and propionylation, as these pathways are increased and linked to fatty acid metabolism, and their increase upon inhibition of EZH2. The EZH2 inhibition improves the tolerance to low glucose or to GLUT1 inhibition, and it increases the maximal and spare respiration capacity of TNBC cells, so it does confer some advantage to the cells. However, the inhibition of EZH2 also creates a metabolic vulnerability to glutamine metabolism inhibition in TNBC. And just to show this to you on a graphic, with a graphic manner, we have cells that are highly dependent on glucose, and they're not sensitive to inhibition of drugs that inhibit the glutaminolysis pathway. However, what we think is that by treating them with an EZH2 inhibitor, we can make them more sensitive to glutamine metabolism and insensitive to glucose withdrawal. And this way they become more sensitive to the inhibition of glutaminolysis. Just to end quickly, I also wanted to highlight the fact that we're also interested in looking at this the other way around, how the cell metabolism can impact the epigenetic reprogramming. And what we notice with this project is that the pools of available alpha-ketoglutarate, for example, and of other metabolites that are used to affect or modulate chromatin modification, they're also affected upon inhibition of EZH2, and this gives a potential for alternative epigenetic marks to be affected upon inhibition of EZH2. And for example, you can see here the drastic decrease in the availability of alpha-ketoglutarate. There's also a decrease in alpha-KG over succinate, which is usually used as a measure of the availability of alpha-KG that is not used in TCA cycle. And we're also interested in other non-canonical epigenetic marks. We've been studying or investigating the succinylation of histone as well with this project. So with this, I would like to thank the people in my lab and also all the collaborators, including Moral Park, Dana Visonis for metabolomic analysis, Simon Pierre Grabel, as well as my former postdoc for this part of the project, Mathieu Lupien. And I'll be happy to take questions.
Thank you so much. This is great work. I really love how you're synthesizing this understanding of epigenetics affecting metabolism, and metabolism affecting epigenetics. It seems like, it's a complex issue, right? And you're really doing great work in that respect. Very well done. So we have a few questions from the audience. The first one is from Kyle Greenland, who asks, have you done any work looking at the expression of the enzymes in the TCA cycle as well as the metabolites? It may be interesting to look at enzymes such as fumarase, which have previously been implicated in DNA repair. Yeah.
So we're definitely doing that. We're trying to understand what gives this specificity. And here I'm not sure I'm answering the question properly, so don't hesitate to let me know if I'm not. But we're looking at how these enzymes are actually contributing to provide the specificity of this regulation, because like we mentioned with Marina's, one of Marina's questions, it's easy to look at this in a global level. However, what gives the specificity of these metabolic change to the chromatin is still not very well understood. And one of our hypotheses is that some of these metabolic enzymes are actually part of some chromatin complexes. And those are things that we're investigating at the moment. We have some projects that are looking at specific metabolic enzymes that translocate to the nucleus in specific metabolic conditions. So it seems to be a very highly regulated process. What we think is that these enzymes might be able to provide local metabolites for the region, the genomic region that's being affected. But yes, we do look also at those metabolic enzymes in the TCA cycle, and fumarate is of course a very interesting one because it's using up succinate. So it's yeah, that's we're pursuing this.
Awesome, thank you. Martin Hurst is asking if mechanistically, do you think EZH2 inhibition is driving K27 trimethylation loss in the promoters of TCA genes in a way that is caused by passive loss? So does it require subdivision, or is it kind of an active mechanism of removal?
Yeah, so that's a good question. We're still not sure about how this is all happening. We think it requires cell division, but there might be some also active loss that's happening. If we think about how this is regulated in terms of metabolism, I mean, the loss could occur because of the activation of a demethylase, I guess. And this is something that I think it can happen both ways, but we're not sure about that.
Awesome. And Talia Barsai-Lovejoy, she's asking if you noticed any regulation of transporter genes, like transporter metabolites.
Yes, so we have transporters of glutamine that are upregulated upon inhibition of EZH2. This is all in cell line. Again, we've started looking at this in tumors, but it's you can imagine how this is a bit more complicated in tumors, especially because of the project that you just saw before with Marina's project. We know that metabolism is not the same everywhere, so we need to keep that in mind when we investigate this. But we do see transporter for glutamine as well as cysteine, and some of the amino acid transporters are upregulated upon inhibition of EZH2.
Awesome. And I have one more question before we end this seminar today. I noticed in the first part of your talk that when you inhibit K27 trimethylation deposition, you're getting double-stranded RNA expression, right? Mm-hmm. Did you follow up on that and see if the double-stranded RNA expression triggered the viral mimicry?
Yeah, so we did, and it's part of it is in the publication. So it does activate viral mimicry. What we did was to knock down some of the genes of viral mimicry and see how they contribute to the effect of UNC, and they did, but not completely. So it's not a full rescue. So this mechanism is contributing to the effect of UNC and the resistant cells, but there's probably something else that's also happening because it didn't completely rescue the effect.
Okay, and then very quickly, there's one more question from the audience. Somebody is asking if you know the expression levels of Jumanji-B3, JMJ-D3 in these breast cancer cells.
If I JMJ-B3, D3?
JMJ-D3.
I think if I remember, in which cells, in the resistant cells?
Yeah, I'm not sure.
Okay, if it's the resistant cells, and I think I remember. Okay, I think I remember this gene specifically. Yes, it's up in the resistant cells.
Okay, awesome. Geneviève and Marina, we really like to thank you for your time today and for presenting your beautiful data. We really appreciate it. Thank you so much.
Thank you.
I would also like to end with a couple of announcements. First of all, I just wanted to thank again Active Motif for partnering with us and supporting our seminar series going forward. Particularly Mason Brooks, as I said before, was really a key contact for us. And I also would like to point out that we have a full slate of speakers for 2022. You can see the speakers and the dates they're going to speak on the new CERC network websites. This is epigenetics.ca. If you haven't checked this website, please go and have a look at it. It's a beautiful website with lots of content, and it's something there is something for everybody, for scientists and non-scientists. So it's something you should look at. On the seminars tab, you'll find the list of the speakers for 2022, and we'll start the year on January 28th. At 9 a.m. Pacific time, 12 noon Eastern time, with a presentation by Jacek Majewski from McGill University. Okay, so we're going to start the year in a great way, and I'm really looking forward to seeing all of you on January 28th. Thank you so much, everyone, and I'll see you next month.
Thank you.